Welcome to our webcast today, everyone. Thank you so much for joining us. I'm Cheryl Rogers, the Director of Marketing here at Golden Helix. Today, Dr. Nathan Fortier, one of our field application scientists at Golden Helix, will take us through a couple of cancer workflows, including a cancer gene panel and a tumor-normal workflow. Nathan?
Hello, everybody. So today, what I'm going to be talking about is cancer gene panels and tumor-normal workflows and how you can go through these workflows using VarSeq. And I'll cover a little bit of what VarSeq is and what kind of features are going to be available to you. So on the agenda for today, we're going to start out by going over who we are as a company. I'll tell you a little bit about Golden Helix and what it is that we do. After that, I'll give a brief introduction to VarSeq and just let you know what VarSeq's really about and what features are available to you through VarSeq. After this, I will go through a demo on cancer gene panels, and I will walk you through the workflow for a cancer gene panel in VarSeq. And then I'll walk you through a workflow for a tumor-normal panel in VarSeq. So Golden Helix is a global bioinformatics company that was founded in 1998. The founder spun off from early work at GlaxoSmithKline, who was a key investor. The company started with a piece of software called Helix tree, which has evolved into the SNP and Variation Suite, or SVS. Since then, we've added two additional products, GenoBrowse and VarSeq. And we have 17 years of experience in the field of bioinformatics, and we've been in the market since before GWAS was born. Here at Golden Helix, we have customers from all around the globe. In fact, we have customers on every single continent except Antarctica. We serve over 350 organizations, including top-tier research universities, clinical labs, government institutions, and pharmaceutical companies. And this leads to thousands of users who trust our software with their labs and research. And our clients have cited us in over 900 peer-reviewed publications. And these include prestigious journals such as Science, Nature, and Nature Genetics. So when you buy a piece of software from Golden Helix, you're getting more than just the software. As a company, we are known for quality products, excellent customer service, and domain experience. Here at Golden Helix, we've been developing software for over 17 years, and we have a wide range of talented experts on staff, including biostatisticians, mathematicians, and software engineers. And we don't just sell products. We seek to understand our customers and their needs, becoming trusted advisors. We are very active in the industry and in the community. We attend conferences where we present reports and demos similar to the webcast that you're seeing today. And we provide training and support from you from the very beginning. And this includes training you on how to use the software and providing support for any help that you would need using the software along the way. And we even can do some custom work for you, including building custom scripts and providing you with access to custom scripts that we've already developed. And here at Golden Helix, we pride ourselves on being very transparent. We openly publish the details about all of our methods, including the algorithms, including the math that's used. And we offer some of the fastest software solutions around. All of our algorithms and software can run perfectly fine on your average desktop computer and can provide results often within minutes. So before we get into the discussion of what exactly VarSeq is, I just want to let you know that if you have any questions at all during this webinar, You can enter those questions in the GoToWebinar window, and we'll try to answer as many of them as we can at the end of this talk. So what is VarSeq? Well, VarSeq is a powerful commercial-grade software designed for local hardware, and it has the largest and most up-to-date repository of public annotations. And we put a lot of pride into getting the details right with these annotations. We provide advanced and powerful filtering through a rich, easy-to-use user interface. And we can allow you to visualize these results using our Genome Browse visualization engine. And all of the workflows that you build and perform in VarSeq can be automatically repeated using our VarSeq pipeline. So VarSeq is ultimately a filtering and annotation platform. And because of that, it's only as good as the annotation sources that support it. And VarSeq is backed by an extensive list of curated annotation sources, and we spend countless hours finding the best sources to curate, and we format these sources to our strict specifications. Before releasing a data source to our customers, we make sure that the data is accurate and represented in a way that can be used for annotating variants. And when we find problems, we go back to the source and fix the issues we find. And we will even fix the data ourselves before making it available on our repository. Additionally, all of your workflows are locked down to specific versions of a specific annotation. And so we will never automatically change the version or update the annotation for you without your permission. You can lock down any of your workflows to any specific version of any annotation in our library. We also offer 2 additional annotations for an additional licensing fee. These include OncoMD, which provides curated drug-targeted mutations for cancer, supporting clinical trials and functional evidence, and OMIM genes, phenotypes, and variants. We also offer an automatic report generation feature in VarSeq that can be used to generate reports from prepared templates. It comes with a standard ACMG standard germline report, but all of these templates are completely customizable. They're written in JavaScript. If you have people on staff that can use JavaScript, you can customize the templates yourself, or we can help you create a custom report template that meets your specific needs. Small changes to report templates are included in the VarSeq reports package, and if you need more extensive changes to any of the report templates, we can perform those as well for a small fee. So ultimately, the motivation for the sort of workflows that we're going to be talking about is just basically the fact that cancer is a disease of the genome. And next-generation sequencing gives us the ability to understand mutations associated with cancer, discover new arrangements in the cancer genome, and it allows us to provide personalized diagnosis and management of cancer. So today we're going to cover 2 types of panels. We're going to cover cancer gene panels and tumor-normal workflows. Let's start by looking at cancer gene panels. So before I get into VarSeq, I want to just give you a brief overview of the data that we're going to be looking at today. We're going to be looking at data from the Illumina TrueSight cancer sequencing panel. This includes 6 reference samples, including 1 HapMap sample, sequenced following standard protocols set forth by Illumina. This data provides comprehensive coverage of 94 genes associated with a predisposition for a number of different kinds of cancer. And we have BAM and VCF files for each of these samples. The samples all have high coverage and average read depth over the targeted regions. For the 6 samples, the average read depth is 574 reads. So with that, let's go ahead and jump into VarSeq and actually look at this product and look at this cancer gene panel workflow. So here we have VarSeq. We have VarSeq open here, and you can see that there are a number of views. To the left, we have a view containing all of the filters that just defines the criteria for including a variant at the end of our what's called a filter chain. And on the right, we have a table listing all of your variants. So you'll notice that this table includes information pulled from the VCF, including information about all your samples, including information about the genomic position, the chromosome, and the start position, as well as the reference and alternate and the identifier. But this table also includes information pulled from a number of samples that VarSeq has annotated our variants against. And this includes information from RefSeq, information from COSMIC, as well as information from OMIM and OncoMD. And in VarSeq, it's very easy to add additional annotations. To do this, you can just click the Add button in the upper left-hand corner of the screen, click Annotation, and you can add any annotation in our extensive public library of annotations to this project. So before we get into the details of the workflow, the first thing we're going to want to do is do some quality control. What we want to do is we want to ensure that there is good coverage for the targeted regions. And if there's not good coverage, we would probably need to rerun our samples. So let's do this quality control by looking at the samples table. And this table is shown at the bottom left-hand corner of the screen. In our samples table, we can see that our BAMs have been linked, that is, our BAM files have been linked to this project. And so we were able to compute coverage for our targeted regions. So the first thing we're going to want to look at is the sample mean depth over these targeted regions. And we can see that for all of these, the sample mean depth is greater than 300. And so the sample mean depth looks very good. Now, if you want to get a more in-depth look, you can also look at the percent covered at 100x. And in this case, you'll notice that between 90 and 100% of the samples are covered at 100x. And so we can safely say that all of our samples meet our quality control criteria. At this point, we can go ahead and walk through this workflow. So we'll start with the very first filter. So these individual boxes here are what are called filter cards. And they define criteria for including or excluding a variant. For example, this first filter card just determines whether or not a variant is in the set of targeted regions. If it is, it's included, and if it's not, then it's excluded. And so here we can see that there are 448 variants that are in the targeted regions. The next filter is just based on the quality control criteria defined by the variant caller. And if it passes the variant caller's quality control criteria, it's included, otherwise it's excluded. The next filter is also a quality control filter. This is a filter on read depth. And we only want to look at variants with high coverage, so we're filtering out variants with the read depth less than 100. The next filter is on alternate allele frequency. And because we don't want to get any false positives, we only want variants that are likely to be true somatic mutations, we're filtering out any variants with an alternate allele frequency less than 5%. The next filter just says that we're only including variants that are located within COSMIC. And finally, we are filtering on effect. And what this filter is saying is that we only want to look at variants that have an effect in the clinically relevant transcript as loss of function or missense. And any of these filters can be adjusted at any time by simply expanding the filter, as we're doing here with read depth. And for example, we may want to look at only variants with a higher read depth than this. So maybe we only want to look at variants with a read depth of 250. And so we can enter this, click enter, and our tables and our filter chain is automatically updated. And so here we can see we have 29 variants that pass these filters. Now at this point, we want to determine which variants we're going to say are primary findings and which variants we want to say are secondary findings for the purposes of including the variants in a report. In this case, we have selected 3 variants, highlighted here in red, as primary findings. And I'll spend a little bit of time going over the reason for selecting these specific variants. So if we scroll over to the right in our variant table, you'll notice that all 3 of these variants have currently recruiting OncoMD clinical trials associated with the genes. And so these variants are definitely actionable. They have a number of drugs associated with them, and these clinical trials provide us with a clear action here for these 3 variants. We've also included a number of variants as secondary findings based on other criteria. And once we selected our variants as primary findings or secondary findings, we can then generate a report. So to do this, we're going to look at this view here called the report view, or I guess the report editing view. And here we can modify any of these fields. We can enter patient information such as the name of the patient. We can enter information about the physician and the institution. And we can also enter information about the specific variants. And so we can provide classification information. And maybe this variant is pathogenic, but maybe we've decided that this one is likely pathogenic. We can also edit any interpretation information. By default, this report populates the interpretation information with information from OMIM. But all of these fields are completely editable and can be changed at your discretion. We also have interpretation information for our incidental variants. And again, all of the fields here, all of the information in the fields is completely customizable, and you can build completely custom reports. Once you're happy with your report, you can click the Create Report button, and this will generate a report. So the first thing to notice about the report is that we've included all the patient information as well as information about the sample. This includes information about the panel coverage and the average read depth. And we calculate this information for you. And believe me, this is a very nice feature because calculating this information by hand is not easy. We also include information about the affected genes, as well as all the information about your primary finding variants. And finally, we include information about the OncoMD clinical trials that are available. We've also included information about the interpretations for all of the variants that has been pulled from OMIM, complete with links to the specific references. And if you scroll to the bottom of the report, you'll notice that we have included all of the references, including links out to the full text for the specific articles for these references. And all of this information has been automatically included in your report. And so this gives you an idea of how you can go from a set of variants imported from a VCF all the way to a report in the context of a cancer gene panel. So the next thing we're going to look at is a tumor-normal workflow. So before we get into the demonstration for a tumor-normal workflow, I'd like to go over the data that we're going to be looking at here in the tumor-normal analysis. So we're looking at exome data from a gastric cancer study. It's an exome pair from this study that was published in Nature Genetics. We've included BAM and VCF files for each sample. The sequencing data was downloaded from the NCBI Sequence Read Archive. And then batch variant calling for single samples was done using BWA plus GATK through the 7 Bridges genomics pipeline. The study for this data set identified 661 genes that contained non-silent somatic point mutations. and this list was used for filtering criteria. So let's go ahead and look at this project and this data in VarSeq. So again, you'll notice it looks much the same as our previous project. We have the same variant table here at the lower right. We have our filters here in the left. You'll notice we have added a new view here in the upper right, and this is our genome browse view. And this allows you to visualize your variants. And we'll get into why this is useful here in a few minutes. But first, let's walk through our workflow and walk through our filter chain. So we start out with a number of quality control filters, similar to the cancer gene panel workflow. We begin by filtering to only variants that pass the quality control filter as defined by the variant caller. Next, we filter to only variants with a read depth greater than or equal to 10. And again, we filter on alternate allele frequency. We filter out variants that have an alternate allele frequency that is less than 1% in this case. The next filter is probably the most significant difference with this workflow, and that is because we have a normal sample, we can actually filter out any variants that are included in the normal sample. And so here what we're doing is we're filtering out any variants that occur in the normal sample with a frequency greater than 0.1%. That is, we only want to look at variants that are practically non-existent in our normal sample. Next, we only look at variants that are in cosmic. And finally, we look at only variants that are in our targeted genes. And this brings us down to a list of only 2 single variants. And so we've included these 2 variants as primary findings for our report generation later on. So before we generate a report, we want to do some quality control. Specifically, we want to verify that the variants are true somatic mutations that are only present in the tumor sample. And we can use our genome browse visualization view to help us with this. So our genome browse visualization is shown in the upper right. And what we're showing here is we're actually showing a visualization of the read depths for our variants. So to look at the read depths at a specific position, we can just click on a variant and it will jump to that position in Genome Browse. At the top, we have the read depths for our normal sample, and at the bottom, we have the read depths for our tumor sample. And so we can look and see if this variant is present in the normal sample. And you'll notice that it's not. So we know that this variant is only present in our tumor sample. And specifically, it's present in the tumor sample at a read depth of 153. Or excuse me, at a read depth of 19. And we can do this for our other variant as well. We can look here and we can see that this variant is not present in the normal sample at all, and it is present in the tumor sample at a read depth of 42. And so once we're happy with the quality here and we realize that these samples do not appear anywhere in the normal sample, we can go ahead and use these variants to generate a report in much the same way we did in the cancer gene panel. We can just enter our information, click the play button, and generate our report. Additionally, if you want to share this information with a colleague without having to generate a formal report, you can actually share the tables themselves by exporting them as a text file, a VCF file, or an Excel file. And once you export these as an Excel file, you can send this Excel file around. It includes all of your annotations with the same color coding you see in VarSeq, as well as links out to the websites for the various pieces of information here. For example, you can link out to your COSMIC data. You can actually link out to COSMIC based on the COSMIC IDs that are present here. And so that gives you a brief overview of what's really possible with VarSeq and how you can use VarSeq to do both cancer gene panels and tumor-normal workflows. If you have any questions that we don't get to today in the talk, you can always email them to info@goldenhelix.com, and you can request an evaluation of the software at goldenhelix.com. Again, if you have any questions, you can enter them in the GoToWebinar window, and we will do our best to get through as many questions as we can today. And that concludes my talk. Thank you.
Thank you so much, Nathan. I've got a quick couple of announcements while people are entering those last-minute questions. Of course, we're recording the webcast today, and I will email the link to the recording and the slides out to you tomorrow. So look for those in your inbox. Our next webcast will be presented on April 6th, and that will present some practical applications of BS Warehouse. For those of you attending ACMG next week in Tampa, please come and say hi to us. We will be in booth number 435, and we've got a lot of great demos scheduled. And of course, after those demos, we'll be giving away our famous Golden Helix t-shirts, so do stop by and say hi. For more specific details on the demos, you can visit our blog, our 2 snips. You can bounce to that right from our website, and you'll get the full lineup. Next week, we will be releasing a 2nd edition version of our cancer ebook, and I'll be emailing those out, so look for those too. And that's a wrap for me, so let's get to those questions. First up here, can you tell me, oh gosh, excuse me, I'm a little tongue twisted. Can you tell me how much you charge to run per sample?
So we actually don't charge at all per sample. The way our subscriptions work is you actually buy a license, and then with the subscription you can run as many samples as you want to through VarSeq. And so you're not limited in the number of samples you can run in any way at all.
Thank you, Nathan. Can you tell me who has implemented VarSeq so far?
So we have sold VarSeq to a number of institutions. I'll just mention a couple of really notable ones. The first is the University of Iowa Cytogenetics and Molecular Lab. We also have a license currently being used by the Rutger Cancer Institute of New Jersey, and this is an NCI designated cancer center. And so we have a large number of customers using VarSeq right now all over the world.
How customizable are the reports generated in VarSeq?
So these reports are completely customizable. You can change pretty much everything about the report by editing the JavaScript for the templates directly. And if you don't have anyone on staff that knows JavaScript, we can create custom report templates for you. We can make major modifications and completely change the templates for a small fee. And if you only need small changes to the existing templates, we can do this free, and it will be included in your VarSeq report license.
Okay, is it possible to export multiple samples at once?
Currently, I'm actually not I'm not aware of any way to export all of the well, actually, so what you can do is you can do an export where you include a number of different tables. For example, here I have an export pulled up. You can actually select which tables you want to export, and you can actually export any number of tables, including the samples table with information about all of the samples. So I hope that answers your question.
Thank you, Nathan. Let me see.
So I know there's a question about Cosmic, and so I can answer that really quick. I know someone was asking, what is Cosmic? Cosmic is just a catalog of somatic mutations for cancer, and they're an organization that keeps track of variants that have been implicated as somatic variants causing cancer. And so when we're filtering on Cosmic, we're filtering to variants that are known to be somatic mutations in cancer as documented by this organization.
Okay. Let's see. Is VarSeq only designated for NGS, exome, or whole genome data?
Is VarSeq designated for? VarSeq can actually be used for a large variety of data. I mean, basically, if you have your data in a VCF format, it can be imported into VarSeq, it can be annotated against, and it can be exported from. And so, basically, any data that can be represented in a VCF from a diploid organism can be pulled into VarSeq.
Okay, next question. Can you show how to link a BAM file to a VCF file and determine coverage?
Sure, I can definitely do that. So if you want to link a BAM file to a VCF file, you do this when you first create a new project. So for example, here I have VarSeq open. If I want to create a new project, I just simply click Create New Project. You select any of these templates that you would like to use for the new project. You then can define your input files. So you can select your VCF files. Once you've selected one or more VCF files, you can actually click this Associate BAM file button here. And from here, you can actually browse to a location that contains BAM files. You can choose the location, and then you can select any of the files that are in that directory as a BAM file. And once you've done that, you can just click Next, and it will automatically associate that BAM file with the specific sample that you've imported. And then it will automatically compute all of your coverage statistics and all of your average read depth information for you.
Do you have any functionality for saving variant interpretation paragraphs that are written?
So actually, that's essentially what's being done when you generate these reports. So if we go here to the Tumor-Normal project, for example, and you go to the report information. If you scroll down here, you can actually enter any interpretation you want to for any of these variants that you flagged. And I mean, right now this information is pulled from OMIM, but you can put any kind of interpretation information you want to in these fields. And in fact, by customizing these reports, we can add any additional interpretation fields that you would like to have if you have specific additional fields for interpretation that you would like to include in your reports.
Do you have any ability to count how many times a variant has been seen in your previous samples and to search for samples that have a specific variant?
So this actually gives me an opportunity to talk about an upcoming product that we're going to be releasing soon. We're here in about a month, we're going to be releasing a product called Variant Warehouse. And this product actually allows you to take these projects, upload them to a server, either stored locally or stored in the cloud if you would like, and you can upload all of the variants from any of your projects to the server, and you can actually search to see if a variant occurs in any sample you've ever seen, in any project you've ever looked at. You can actually have this link out to the report for that sample, and so it really gives you the ability to search over any sample you've ever seen for information about a specific variant.
Okay, I think that about wraps up the majority of questions. There are a few lingering that I will get in touch with you regarding those. I want to thank Nathan for your time today. Great webcast. I want to thank everyone out there for coming as well. If you're interested in taking a look at VarSeq or VS Warehouse for that matter, feel free to get in touch with us. You can pop on our website and get more information there, or you're welcome to email us at info@goldenhelix.com. Thank you again, Nathan.
Thank you.
Have a great day, everyone.